In this session, we will study about the cytoplasm and the cytoplasmic organelles. Okay? In our previous session, we have already discussed about the structure of the cell and why the cell is known as the unit of the life, basic unit of the life. And in that session, we have discussed about the plasma membrane, cell wall and nucleus. Okay, now we will carry forward with cytoplasm. Okay, now the dense liquid fluid inside the cell membrane, it is known as cytoplasm. Okay, the fluid content inside the cell, the plasma membrane, is called as the cytoplasm. Okay, it contains many specialized organelles. In our previous session, we had talked about division of liver. So, irrespective of the cell, whether it is unicellular or multicellular, dear students, what is there? There are certain specialized organelles to perform various or different type of the functions. Okay? So, there are a number of specialized organelles performing a specific function. Means that individual organelle will perform an individual function while a different organelle will perform different function. Okay? Now, as we discussed that nucleus or the nuclear membrane, they take the dense stain, while in the case of this cytoplasm, it takes very little stain. Okay? Now, we should remember, as we have already discussed about the differences between prokaryotic and eukaryotic cells. Dear students, the membrane-bound organelles are absent in the prokaryotes. Okay? So, we will see it later on that ribosomes are the organelles which are found only in the prokaryotes. Of course, they are found in the eukaryotes also. But dear students, we have to keep in the mind that in the case of prokaryotes, the cytoplasm, they are devoid of the cell organelles. Okay, most of the cell organelles. Okay. Now, again we have made it very clear that eukaryotes have membrane-bound organelles and a well-defined nucleus. Okay. Now, what we will see. Now, what is about the COVID-19? Okay, dear students, nowadays there is a very much panic about the COVID or corona. Okay, what is that COVID-19? Dear students, it is the virus. Okay, virus, as we have already discussed, they are the interlinks between non-living and living things. Okay, so why we say that it is an interlink between non-living and living? So at this level, dear students, this is just because of the membrane. Okay, it is the significance of the membrane. Okay, now what is there? That viruses lack any membrane and hence do not show characteristic of life. Okay, so we have seen in our previous session that any cell, it is bounded by a cell membrane, and in the case of plants, dear students, it has got cell wall also. Okay, so viruses, they lack membrane when until they have entered a living body. Okay, living body or host. In that it behaves or acts like as a living being. Of course, it uses the machinery of the living host, and otherwise it is known as non-living. Okay, and use its cell machinery, whose machinery? The machinery of the host to multiply. There, that's why they are the connecting links between non-living and living beings. Okay, now we will see about the cell organelles. Okay, so as I mentioned you, a prokaryotic cell or eukaryotic cell or a simple cell or complex cell undergoes a lot of chemical activities. Whether a cell is unicellular or multicellular, or it is prokaryotic or eukaryotic, it undergoes a lot of chemical activities. Metabolism. Cell may be undergoing the biochemical activity of synthesizing, that is anabolic, or it may be undergoing the catabolic pathways, and to support their complicated structures. Okay? Complicated structures and functions. What is that? Just suppose any cell is undergoing mitosis, then again it has to be synthesized, the cell membranes or cell wall in the case of the plant. So these are a number of activities which a cell performs. Okay? Now, to keep different activities separate from each other, the cell has membrane bound little structures. These are very minute structures, and some of them can be visible only under electron microscope. Okay, so these structures, they are known as cell organelles. Okay, some of these organelles are so minute, as I mentioned just now, that they are visible under electron microscope only. Okay, so some of the organelles which will be studied today, that is endoplasmic reticulum, mitochondria, ribosomes, plastids, and vacuole. Okay, so we will pay attention on the cell structure, or the structure of the membranes, what is the composition of the membrane and what functions they are performing. Okay, so now we will study the first organelle, that is endoplasmic reticulum. Dear students, in short we can write this as ER. Okay, so endoplasmic reticulum, or ER, E for endo, R for reticulum. Now, dear students, it is the large network of membrane bound tubules and sheets. Okay, it is a membrane bound tubes and sheet. This is structure. It's a complex structure. I will just show you in the diagram. Okay, it looks like a tubule or bag. The shape may be round or it may be oblong and varies in appearance in different cells. Okay, the shape, the size and the structure of endoplasmic reticulum, dear students, it varies from cells to cells. Okay, now you should keep this thing in the mind, that the membrane of this endoplasmic reticulum, dear students, it is lipoproteinaceous in nature. That is, it is made up of lipids and proteins. Okay, now endoplasmic reticulum, dear students, it serves as a channel for transport. It is meant for transportation of materials, especially proteins. Okay, where between various regions of the cytoplasm or between cytoplasm and the nucleus. Okay, now dear students, endoplasmic reticulum, it behaves like a channel of transportation between the cytoplasm, between the nucleus and the cytoplasm, and dear students, as the materials are transported towards the membrane, and thus they can move across the membrane also. Okay, now what is that? It also functions as a cytoplasmic framework. It works like a framework. It provides a surface for some of the biochemical activities of the cell. Okay, now what I told you, that it behaves like a framework and it is a site of activities of the cells also. Okay, so we are discussing about endoplasmic reticulum. Okay, now the next thing is that, as I told you, it is a structure of the endoplasmic reticulum. Dear students, as I told you, it is a vesicle or tubular structure consisting of the vesicles. Okay, so it is a structure of the endoplasmic reticulum. Okay, now you are seeing the beaded structure on the wall of this endoplasmic reticulum. So this is our next topic. So when endoplasmic reticulum is having these ribosome molecules, ribosomes are also the cytoplasmic organelles, and dear students, I just mentioned you that ribosomes are also found in prokaryotic cells. Okay, so when they are associated, when we observe on the surface of the endoplasmic reticulum, so this type of reticulum, endoplasmic reticulum, it is known as rough endoplasmic reticulum. Okay, now we will see that we have differentiated this endoplasmic reticulum into two types. That number one is when it is known as rough endoplasmic reticulum, as I mentioned just now, that when ribosomes, the cytoplasmic organelles, are attached to the surface of the endoplasmic reticulum, as we have seen in the diagram. So this type of endoplasmic reticulum, dear student, it is known as rough endoplasmic reticulum. Okay, so ribosomes, again these are the minute, and mind it, they are having a single walled structures. These are also found in the prokaryotic cells. Okay, so these ribosomes we will study it later on. These are the sites or manufacturing sites of the proteins. Okay, the proteins are manufactured and are sent to various places. Okay, the protein synthesized in the ribosome, they are sent to various places through this endoplasmic reticulum. Okay, now what is the significance of this endoplasmic reticulum? So dear students, dear students, as I told you that in contrast to rough endoplasmic reticulum, when endoplasmic reticulum is not having, or the surface of the endoplasmic reticulum is without ribosome, so such type of endoplasmic reticulum is known as smooth endoplasmic reticulum, or SER. S is for smooth endoplasmic reticulum. Okay, now it helps in the formation of fats and lipids. Okay, dear students, it helps, which one? Smooth endoplasmic reticulum. It helps in the synthesis of fats and lipids. And now the very important role played by SER is that in the detoxification of many poisons and drugs in the liver cells of vertebrates. You know, the organisms which are having the vertebral column, they are included in vertebrates. And dear students, this smooth endoplasmic reticulum, it helps in the detoxification of many poisons and drugs in the liver cell. Okay, now this smooth endoplasmic reticulum, as I just told you, that it is the site of membrane biogenesis. Membrane biogenesis means that the synthesis or the building of the cell membranes. Okay, so whatsoever the membranes of even the cytoplasmic organelles. Okay, so dear students, the membranes of cytoplasmic organelles or even of the cells, it plays this, which one? That endoplasmic reticulum, and particularly the smooth endoplasmic reticulum plays a significant role here. Okay, now our next organelle is the Golgi apparatus. Okay, so the second our cytoplasmic organelle which we will study, dear students, it is Golgi apparatus, and it is also known as Golgi bodies, as it was discovered by Golgi. Okay, so now as like endoplasmic reticulum, dear students, Golgi apparatus, I will just show you. Golgi apparatus is also a complex cellular system. Okay, like endoplasmic reticulum, Golgi apparatus is also a complex cellular structure. Okay, now it consists of membrane-bound vesicles. Okay, it also consists of membrane-bound vesicles, how arranged approximately parallel to each other. Now what we will see that these vesicles, in the case of Golgi apparatus, dear students, they are arranged parallel to each other in stacks called systems. Okay, so what these stacks of these Golgi apparatus or vesicles, they are known as systems. Okay, so these are the systems of Golgi apparatus. Now, what is the role of Golgi apparatus? Dear students, now pay attention. It stores, modify and package materials in vesicles. Okay, synthesized near endoplasmic reticulum. So whatsoever the material or biochemical synthesized, they are stored, they may be modified, they are packed inside the vesicles of Golgi apparatus. Okay. Now, in some cases, simple sugars, simple sugars are converted to complex sugars in Golgi apparatus. Dear students, when you will come in the higher classes, at that time we will tell you that the simple sugars, they are the monosaccharides, disaccharides and trisaccharides are the some polysaccharides. These are the types of the sugar. So simple sugars, okay? So simple sugars, they are converted to complex sugars in the Golgi apparatus, okay? Now, the next thing is that the material formed in endoplasmic reticulum are also dispersed in and outside the cell through Golgi apparatus. Okay, so as we have seen that endoplasmic reticulum, it was providing vesicular structure. So like Golgi, in the case of Golgi apparatus, dear students, the materials formed in the endoplasmic reticulum, they are dispersed in and outside the cell. Okay, it was endoplasmic reticulum was the medium of transportation inside the cell. So outside the cell, it is achieved through the Golgi apparatus. Okay, now our just see the diagram of this Golgi apparatus. As we have mentioned that it is a vesicular structure known as cisterns. So likewise, just see all the structures. It is the parallel structure, and these parallel structures of the vesicles, they are known as the systems. Okay. Now, coming to the next point, our next organelle. Just see this is Mr. Golgi who has discovered that. And dear students, I would like to tell you something about Camillo Golgi. Okay, now you will have certain questions from Golgi, Mr. Golgi or Camillo Golgi in your NCERT examinations, dear students. Now see, the important discovery was about the structure of the description of the structure of Golgi body, and hence we call that Golgi apparatus as Golgi bodies also. Dear students, Camillo Golgi worked on nervous system. Basically, he made research over the nervous system, and secondly, the role he plays is in about the staining of the nerve cells. Okay, so nerves, they are very complicated structure, and that is known as black reaction. Okay, so staining of the nerves and cell structure is known as black reaction. And what he did there, so he used a dilute solution or the very weak solution of silver nitrate for staining and ramification of the cells. Okay, so cells when they were dividing and redividing and proliferating here and there, so we can stain those ramified cells with the help of this silver nitrate solution. Okay, and as I mentioned you that Golgi discovered and described the Golgi apparatus. Okay, now our next cytoplasmic organelle, dear students, it is mitochondria. In your previous classes, you have already known about the powerhouse of the cell. Okay, you have learned, you know that all the cells, all the tissues, all the organs, they require energy, and the powerhouse of the cell, it is, here students, it is the mitochondria. Okay, again it is a double membrane structure. Okay, mitochondria is a double membrane structure. The outer membrane is smooth. Okay, when you will see the structure like that, so outer membrane it is smooth, while the inner membrane it having the deep foldings. Okay, so it is the deep, having the deep folding. I will just show you the diagram. And this structure is known as cristae. Okay, so there in the case of Golgi bodies, dear students, we have seen the parallel vesicle-like structure. They are known as cisterns, but the inner foldings of this mitochondria, they are known as cristae. Okay, the foldings of inner membrane cristae create large area for ATP generation. Okay, ATP, adenosine triphosphate. It is an energy-rich compound. It is generated inside the mitochondria, and that's why mitochondria is known as powerhouse of the cell. Okay. Now, the one of the characteristic property of the dear students mitochondria is that, as we will see later on, that this mitochondria, with some other organelles also, they are having their own DNA. Dear students, it is a typical organelle along with plastids, as we will discuss just later on. So they are having their own DNA and ribosomes. Okay, so the ribosomes present in the prokaryotic cells, they are similar in the which are found in the mitochondria. So you have to remember this thing. Now, mitochondria, as I mentioned, are the sites of ATP, adenosine triphosphate, the energy-rich currency of the cell. It is secreted in the mitochondria. I mentioned you, it is the inner membrane of the mitochondria, and these enfoldings, they are known as mitochondria. Dear students, mitochondria are known as the powerhouse of the cell. Okay, are you getting what I am saying to you? Now, our next important organelle, dear students, it is the lysosome. This is also known as the suicide bag. Okay, so lysosomes are the membrane-bound sac-like structure. Again, they are sac-like structure. Now, keep this thing in your mind. They are filled with digestive enzymes. Okay. These lysosomes, they are filled with digestive enzymes secreted by RER. R means rough endoplasmic reticulum. So these lysosomes, they are the membrane-bound structure having the digestive enzymes secreted by rough endoplasmic reticulum. Okay. So what is the role of lysosomes? Dear students, this lysosome keeps the cell clean. Okay, so what they did, any foreign material, bacteria, worn out cells, or all organelles. Okay, so old organelles, worn out cells, bacteria, or any foreign material which has come inside the cell, it is degraded by the digestive enzyme activities of the lysosomes. Okay. Now, one thing again we have to keep in mind, during disturbances in cellular metabolism, lysosomes may burst and the enzyme digests their own cell. What happens? That's why we call it as the suicide bags. Why we call suicide bags, dear students? During disturbances in cellular metabolism, lysosome may burst. They burst and they secrete the enzymes, the digestive enzymes, and thus harms the cell itself. That's why they are known as suicide bags. We will continue with our next cytoplasmic organelle, that is known as plastids. Okay, you have heard about the chloroplast, chromoplast, and leucoplast. Okay, so these together, leucoplast, chloroplast, and chromoplast, dear students, they constitute plastids, and basically we characterize or differentiate this plastid into, categorize into two. One is the chromoplast. Chromoplast means the colored one, and leucoplast, that is the white one, because, and the next one is the chloroplast. Of course, it is green in color, but dear student, as you will study in higher classes, that the chlorophyll along with this chloroplast, along with the chlorophyll also contains some yellow and red color carotenoid pigments, and that's why we differentiate this plastid into two types, that is the chromoplast and the leucoplast. Okay. Now internally these plastids they are consist of many or numerous cell embedded cells which are filled in the embedded in the fluid that is known as the stroma. Okay, so plastids, as I mentioned you, they consist of numerous cells, and these cells they are embedded in the stroma. Okay. Now, the imaginations of the cell from the thylakoids or the stacks like grana. Okay, I will just draw a structure. You just see here. If you will visualize the chloroplast, so it is a structure like that, and they are again the membranous structures having the stacks of the coin-like structures. Okay, so having one over the other. So they are known as the grana, having these thylakoid membranes. Okay, so it is the structure like that. So as I mentioned, like mitochondria, dear student, these chloroplasts or these plastids, they also have their own DNA. They also have the DNA and ribosomes. Okay, and now as I mentioned, that plastids they can be differentiated or categorized into three types: chromoplast, leucoplast, and chloroplast. Okay, so these are the very important organelles and basically defined in the green plants. Okay, now last we will study about the vacuoles. So what are the vacuoles, dear students? Cells secrete varieties of the substances. Some may be enzymes, some may be carbohydrates, some may be proteinaceous in nature. So depending over that, they store in different types of the vacuoles. So they are single membrane structures containing various vacuoles. Vacuoles store, as I mentioned, to solid or liquid contents of the cell. So whatsoever the content of the cell, dear students, they are stored in the vacuoles. Okay, so they may be amino acids, they may be sugars, various organic acids, as I mentioned you, proteins, etc. Okay, now as we have learned in our previous session that what is the difference between the animal cell and plant cell, dear students, I have already mentioned that animal cells have small vacuoles and compared to the plant cells. So whenever anybody asks you about the differences between animal cells and plant cells, then you have to mention this thing, as it is the important thing, that animal cells have small vacuoles as compared to the plant cells which have the large vacuoles. Okay, and in some of the plants, dear students, they occupy 50 to 90% of the volume. So in this way, dear students, we cover about the cytoplast. I told you about mitochondria, I told you about plastids, I told you about endoplasmic reticulum. In endoplasmic reticulum, we saw that we categorize it into smooth endoplasmic reticulum and rough endoplasmic reticulum. We discussed about ribosomes, the organelles which are also found in the prokaryotes and eukaryotes, and later on we saw the Golgi bodies. We saw the achievements of the Mr. Golgi. Later on we learned about the vacuoles. Okay, now have you got it? Have you understood it? Now we are going to have some question answer section. Okay, in this question answer section, I will simply put the question and I will give you the answers in my next. I will add these questions in answers in the next video. Okay. Now you have to give the answer. You have to fill the blanks. Okay. Cells. My first question, dear students, pay attention. Cells not having nuclear membrane. Okay, we have already discussed this in the previous session. Having not nuclear membranes are known as. You have to give the answer, and I will give the right answer in my. I will tell you when I am going to give the answer. Okay. The second question is cell membrane is made up of. Okay. What is the composition of the cell membrane? Dash 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 is synthesized in ribosomes. What is the color of plastids? Sorry, white color plastid is, which one is the white color plastid? You have to answer it me. It is known as the powerhouse of the cell. Okay, white color plastid, I have already told you, the infoldings of the inner membranes of mitochondria is called. So what we call the infoldings of the mitochondria ammonia. Okay, in the same way, Golgi apparatus was discovered by. Now, photosynthesis takes place in this organelle. You have to tell me the small vacuoles are found in which cell, whether in the plant cell or in the animal cell. Okay, now, next I have put some these multiple chat questions are there. Which one is a single membrane structure? Lipid molecule is synthesized here. The only cell organelle found in prokaryotes. The cell wall in plants is located where? Okay, so these are the questions. You have to give me the right answers, and I will see that who is going to give me the right answer. Or in subjective way, you have to differentiate between plants and an animal cell with the diagrams. What are the differences between prokaryotic and eukaryotic cells? We have already discussed. What is osmosis? And dear students, you just describe along with the structure and functions of these organelles: nucleus, mitochondria, endoplasmic reticulum, Golgi bodies. Okay, have a good day. Bye bye.